growing up, I was always super fascinated by the ocean and marine organisms. And I was so obsessed with going out and looking for different things on the beach, different jellyfish that washed up. But I didn't exactly know what that meant. When someone said, "What do you want to be when you grow up?" I didn't really know. I know that whenever I was a toddler, I told my mom that I wanted to be a fish doctor. So she kind of took that as a marine biologist. But I really wasn't sure. And when I got to high school, I got super interested in biology, and specifically cell biology. So I thought that I would be more along the medical path. And then when I got to college, I started that path and I did some volunteering. And then I came across this lab that looked at cell biology, but in marine organisms. And specifically, the organism that I work on today, that's the organism that I worked on in undergrad. And that's when I realized that I really liked research, and I really liked the way that it was set up, and I liked the flexibility, and I realized that I really wanted to pursue that. And that's how I got to where I am today, was going from my undergraduate lab to the lab that I work in at the Whitney Lab. My name is Dorothy Mitchell. I'm a cell biologist in training at the University of Florida's Whitney Laboratory, where I study ctenophore regeneration. For most laboratory scientists, their animals are raised over several generations and maintained in the lab. But for me, my model organism needs to be caught from the wild each time I want to collect their embryos. The ctenophore, otherwise known as a comb jelly and regularly mistaken as a jellyfish, is a marine carnivorous jelly that likes to prey on everything from mysid shrimp to fish larvae. The most characteristic feature is the giant ciliary combs that aid in movement. The cilia within these combs act as a prism and break visible light into its components as they are beating. This produces a beautiful rainbow as light hits it. The spectrum of color is a huge help when trying to spot them in the water. Luckily, I work by a great spot on the Atlantic coast where these marine animals can be found. As you can see, it's pointless to look over on this side because it's so choppy. The current is coming straight in from the Atlantic through the St. Augustine Inlet. However, we like to look over on this side because the water is glossy and the current from the other side actually pushes the animals under the dock and up to the surface where I can see them. The only problem is trying to catch them before they drift out that way. We got lucky because a day like today, it's super sunny. This direct sunlight makes it much easier to see things in the water than it would be on a cloudy day. This is also a great spot for seeing other wildlife. After an hour or two of hunting, I count up all of my animals that I've collected in the bucket, make sure they aren't overcrowded, and grab my equipment to pack up my catch and head back to the lab. Once I have arrived at the Whitney Laboratory, I immediately take the buckets into the wet lab room. This room contains the seawater filtration system, allowing me to collect cleaned, full-strength salinity seawater directly from a hose. The salinity of the seawater at the collection site should be equal to the seawater from our system. To ensure that the animals can comfortably be transferred to our water, I'd spend some time placing small amounts of the system's water into the bucket from the collection site. I do this for about an hour, then I set up several buckets filled with the system's seawater and carefully transfer the animals from their collection bucket into the fresh seawater. The animals are now in the buckets that they will reproduce in. Ctenophores are hermaphroditic, meaning that they will produce both male and female gametes and can reproduce on their own. Due to their circadian rhythm already established in the natural environment, these animals will spawn at around 3 a.m. the morning following collection. I will collect the embryos from the animals I caught today tomorrow morning. Once the adults have completed spawning, they find their new home in these large chrysal tanks in the wet lab. These tanks have a constant input and output of fresh seawater. This flow of water creates a circular current that gently pushes the ctenophores around the tank so they do not crowd in one area and foul the water around them.
It's time to head back to the lab I work in. My graduate advisor is Dr. Mark Martindale, director for the Whitney Laboratory and experimental embryologist. The Martindale lab primarily focuses on using experimental techniques to answer questions about patterns in development and regeneration. We have a group of 8 scientists who work as technicians, postdoctoral associates, or graduate students. At any given time, there are numerous projects going on, but we are constantly collaborating on ideas and methodology. Since the animals I collected today will need overnight to spawn, I will not get any embryos from them at this moment. Luckily, I followed the same procedure the previous day, so I have thousands of small embryos to collect from animals that spawned last night. To collect these, I immediately remove the adults from the bucket and use a large pipette to put them into clean water. When I remove the larvae, I place them into small glass finger bowls filled with fresh water so that they can continue to grow. Over the days following the spawn, I will feed these larvae until they are at the desired size for my experiments. Now that I know the experiment that I'm going to do, I need to do some background research to understand the concepts. As a student, it is really important that I understand what is actually going on in the experiment while I'm using it, so I can make the correct biological interpretations. What I normally do is spend some time looking up publications online and brainstorming different modifications I might need to make for my animal. I take the adults that I collected from the wild, bring them into the lab to spawn, and grow up their embryos into larvae. These larvae are called cydipids. They have a slightly different morphology than the adults, but they are capable of feeding and regenerating. We refer to the portion of the body closest to the mouth as the oral end and the opposite side as the aboral. My collaborator, Dr. Allison Edgar, a postdoctoral researcher in the Martindale lab, takes these live cydipids and performs surgeries where she cuts off a chunk of the tissue on the aboral end. This tissue is discarded and regeneration can now be monitored in the cut animal. We need to identify which cells are actively replicating in our regenerating animals. We perform the same experiment on uncut cydipids to separate the normal pattern of cellular division versus changes that occur when animals have been wounded. First, we place the cut and uncut animals into solutions containing the molecule EDU. This molecule is an analog of the thymidine molecule, a nucleoside found in DNA. EDU and thymidine only differ by one component in their structure. EDU differs by the presence of the structure called an alkyne group. Because the EDU molecule is so similar to thymidine, it will be added to the newly synthesized DNA strands in place of the thymidine nucleoside. We are left with newly synthesized DNA strands that contain EDU instead of thymidine. We now have successfully added EDU to the DNA inside the nucleus of the cells that we are interested in. There is no way to visually differentiate the EDU-labeled cells from the others at this time. We will fix the cells by placing the animals in alcohol to preserve the tissue. Conveniently, the alkyne group that differentiates the EDU from the thymidine is highly reactive with the fluorescently tagged azide molecule. We introduced the fluorescent azide solution to the fixed animals, and now our cells of interest contain DNA that will fluoresce. We can now image the animals on a microscope that uses lasers to excite the fluorescent azide on the EDU-incorporated DNA. We now have our data. This image to the left shows an uncut cydipid with nuclei labeled in blue and EDU labeled in pink. As you can see, there are some areas in the animal that undergo constant cell division. The image to the right shows the EDU pattern in animals put in an EDU solution 24 hours after surgery. Here we can see EDU-labeled cells are on the wound site on the aboral end of the animal. There isn't a clear designed outline for which experiments I will do during my graduate studies. Some days I do several experiments, and some days I don't do any at all. One experiment's findings will lead to another, so it isn't exactly predictable. I think that being able to explore different techniques and make your own path toward discovery is one of my favorite aspects of research.
A lot of times I come home and I'm done for the day. I don't do any other work, but some days it's really important for me to continue doing reading or writing or following up on some sort of technique that I'm working on. And I find that most of the time I'm really excited to do these things, because when I do feel like working at home at nighttime after being in the lab, it's mostly because I'm really excited about something that I found the previous day or something I really want to follow up on. And I think that's what I love about science, is that you're constantly discovering new things and you're kind of creating your own path towards discovery. There's not a set curriculum for how you should do things during your graduate study. Your advisor is there to guide you and help you towards specific goals, but you're kind of figuring out these things along the way as you're doing different experiments. So whenever you're done with everything, everything will be uniquely yours and completely original. And likely a lot of the things that you have done and the things that you will find are things that no one has ever done before. And I think that's really cool. And that's what makes me want to keep going, and that's what makes me love it so much.